Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy, and Sarantis.
Welcome to the Proteomics in Proximity podcast, and today we're going to be talking about HATTR and amyloidosis. What is that, Cindy?
So we're specifically, we've narrowed down this really I think a fantastic paper from Samina Ticau and Paul Nioi, who looked at it looked at essentially a broad proteomic profiling of samples that they just had sitting in their freezers. And so, Evan Mills on our team has often said that these samples that have completed clinical trials and are sitting in the freezer might have some nuggets that we might be able to mine by doing a broad scale approach in proteomics. And that's exactly what this team did. And they came out of it with a biomarker that shows potential for not only diagnosing a hard to diagnose genetic disease, it shows promise as monitoring disease progression as well as response to the therapy that was part of that clinical trial. So yeah, this is an exciting paper.
So to back up a little bit, what is HATTR? This is a rare disease, I understand.
Yep, that's right. It's a hereditary Transthyretin-Amyloidosis. So the TTR is transthyretin gene, which is a protein. So there's a mutation in the protein for TTR, and it results in some amyloid. We know amyloid from our discussions around plaques, particularly in Alzheimer's disease.
Correct.
Yeah. So here you have these aggregations of proteins that result in some challenging polyneuropathy that is progressive and fatal. So you can imagine being someone who's been tested for this and they find out they have it and they're just got a ticking bomb. They just got to wait and test and wait to see if it shows up, if they ever evolve. It does. It sounds awful.
Because this is a progressive debilitating disease because it affects your nervous system, but it also affects other organs. Is that correct?
It affects the as I understand it, it affects the ability of the Schwann cells, I think, to give their signals. I think it's interfering with the signals in the musculature. It sort of sounds. I remember
my high school physiology. Let's see, Schwann cells is one of the types of nerve cells. Is that correct?
Yeah.
In sending signals.
Yeah, I remember reading something about the proposed mechanism of the disease, and of course, as most disease. There's heterogeneity, which makes it challenging also to stratify and get through a successful clinical trial.
I see, and thus the need for this kind of study. Can you describe what they did?
Yeah, sure. So they did a cross-sectional arm as well as a longitudinal arm. So essentially, they essentially looked at their cases where they're giving the therapy over at baseline at 9 months and 18 months, and they looked at their placebo, and it was randomized, double-blind, I believe, and that was the component that was longitudinal. So again, 3 time points over the course of treatment, and then they had a cross-sectional aspect of just healthy controls that they monitored over that same period of time.
So this is a clinical trial that they were conducting, like phase 2 or phase 3?
Yeah, I presume it was phase 3.
Phase 3, yes, it's phase 3.
Yeah, it looked like a phase 3 trial. I don't remember them stating that in the paper, but they probably did. It's called the Apollo trial. It's a pretty well-known trial because it's an RNAi therapeutic. So it's an interfering with RNA.
So RNAi therapeutics, help me with this. Yeah. Is that like gene therapy? Is this like a vaccine? We've talked about mRNA vaccines. Is it like that?
There are very few. To my knowledge, there are very few drugs that are FDA approved for double-stranded RNA that target the silencing of the gene. At the end, what happens with this let's say, pathologies, we have a mutated version of the TTR protein and also a wild-type version of TTR, kind of a overexpression of the protein. And in that case, they try to target the expression of the mRNA post-transcriptionally at the cytoplasm. They inject the double-stranded RNA, target the 3' untranslated region of the gene. This is the RISC complex with the dicer argonaute that helps the targeting actually of the antisense strand on the complementary region of the gene, and then degradation of the mRNA with Argonaute 2. That is the endonuclease of the system, actually. So that's pretty much the mechanism of action.
Okay, so with dicer and Argonaute, understood.
Absolutely.
But backing up a little bit, right, these COVID vaccines, right, from Pfizer, BioNTech, and Moderna, I mean, these were mRNA vaccines that were used to actually produce spike protein. But here, what you're talking about RNAi is similarly, right? They're injecting RNA.
They inject double-strand RNA targeting the 3' untranslated region of the gene.
Interesting.
That's pretty much what happened.
And then it actually then turns down the expression of the
gene.
And they're tackling the mutant form, is that correct? The one that's messing things up?
They're tackling both, because this region that they are targeting is for both the mutant and the wild type. And they target both. They don't regulate both of the
But the disease problem,
as I said, patients they have two alleles, one the mutant and the other the wild type. Usually, most of the cases.
And the whole problem is it's just overexpression.
Kind of, right? Because you have the overexpression of the mutated version as well, right? You have the presence of the wild type and the presence of a new protein that is misfolded and aggregated.
I see. So then they developed this drug. What is this drug called?
Patisiran.
Okay.
Patisiran.
So Alnylam produces this drug, Patisiran, but this drug actually is RNA for RNA interference. And we're talking, Cindy, then also about the middle of this clinical trial, and they're using healthy versus affected patients. Then what did they find?
Yeah, so they, in their cross-sectional arm, they found that an expected protein, and the longitudinal arm, so in the aspect without the healthy controls, so just looking at the placebo and the treated over baseline 9 months and 18 months, so in that longitudinal arm, they looked at the time progression of different proteins as patisiran was given, right? So they're looking for signals in various proteins that are popping up above significance for being differentially expressed. And they found 2 of note. They found, I think, 66 total that were providing significant differences, but the 2 that really popped out, and especially in those volcano plots, are NT-proBNP, which was an expected signal, and in fact, I think that was part of the clinical trial using NT-proBNP. But what was unexpected was NFL, and of course, that's part of the title of the paper. Neurofilament light chain is a biomarker of hereditary transthyretin-mediated amyloidosis. And so with NFL, they were finding that the placebo group progressively increased in their NFL levels over the course of the 18 months, and that they could compare. So once they got those signals in the longitudinal arm, now they could actually put in the NFL levels for the healthy controls and look at healthy controls relative to those placebo treated. Those are going to be the ones that aren't going to be getting any better. And they're going to be comparing the healthy controls to those that are treated with patisiran. And what they saw was that the placebo samples were increasing in neurofilament light over the course of the trial, whereas those treated with patisiran were trending toward healthy controls over the course of the trial.
Now, as far as then the results of the trial, the people that were on patisiran got better or didn't get worse. Is that correct?
That's correct. And in fact, there's a statement around the current methods by which we monitor for progression of polyneuropathy in these patients, or in even initial diagnosis for these patients. And so there's a neuropathy impairment score that they used. And in fact, they used this to stratify and figure out who are the patients that are relevant for the clinical trial. You want ones that have, active disease, obviously. And they were able to see that that the impairment score improved as the neurofilament light levels went toward healthy controls. So that's a nice suggestion that that you might be able to use neurofilament light to actually identify when that impairment is happening. The challenge with, I think, that impairment score is it was described as burdensome to administer. I'm guessing, with some of these scores where they're doing a movement requirement, they can be a bit objective, hard to really nail down across different doctors' offices. I'm just making that assumption that that's what they mean.
You're talking about a subjective measurement of some ability to do something, right, in terms of nervous activity. And here, backing up, how did they discover neurofilament light? I mean, this is using Olink panels then, yeah?
Yeah, good point. So we should probably emphasize that a little bit. They used our tool that was the broadest look at the proteomic at the proteome that we had at the time. So they essentially used all of the unique proteins we had in our library across, I think it was 13 or 14 different panels. One of our 14 human panels is actually redundant with the others. So I think it's 14.
This is an Olink Target 96.
Yeah, 13. Yeah, exactly. qPCR panels. And so each of those qPCR panels have 92 proteins along with controls. So you can run 96 samples against 96 proteins at a time, including those controls. So they did a broad look, as broad as they could with what was available at the time. I think it would be interesting to see what they could do now with just under 3,000 proteins, right? We've more than doubled the library since then, and on the NGS readout.
Right. So out of these, over 1,000 proteins, they were able to, you mentioned, find 66, and you mentioned the two of note. The NT-proBNP sounds familiar. Isn't that a cardiac marker?
I would ask the man who is the specialist on the affairs of the heart.
I think also could be a cardiac marker as well. Could be cardiac as well, it's true.
So why do they find, Cindy, you mentioned that the proBNP was expected. I guess that was already known with this amyloidosis condition.
Yeah.
Any thoughts why you have a marker for heart health that crosses over to basically neurological disease?
I think that happens a lot, right?
Oh,
I think
really?
that's where the excitement is where, our bodies are incredibly good at repurposing different things that, we all thought that there would be this really simple smoking gun in the genetic world, right? Once we were able to look at genetics, we thought that we would have one mutation for each disease. And now we find that there's actually lots of different mutations that lead to, death by 1,000 cuts for risk for various diseases like cardiac disease and stuff like that. So I think the overlap of how these different diseases have the same pathways is going to tell us something about mechanism. But I think the speculation, I don't remember if they speculated in the paper, Dale, but I'm guessing you might have some ideas, but I would push it over to our Cervantes affairs of the heart.
I'm taking the difficult part. I don't know. I mean, I have seen a little bit in the literature that this amyloid could be formed also with different tissues like cardiac, and people they have a combination of disease like neuropathy, cardiomyopathies, and usually these amyloids, they target cells that they are not mitotic active, like cardiac cells.
Interesting.
Like, they are polynucleate and they are not mitotic active. Usually they target these. And
fascinating
could be an explanation, but of course they don't they don't go through the details and they don't try to give an explanation.
Yeah, I think they just stated as fact, right, that this was not a surprising finding. The fact that they got not only these two markers, but another 64 of them, that seems to be quite a lot of markers, right? They're not going to chase after each of them. But what can you say about, naturally the title is around one marker. Is there value in looking at more than one?
So I'll just jump in and say this group is part of the UK Biobank Pharma Proteomics project now. So they joined in kind of a second wave of additions to that proteomics project. Initially, it was 10 partners. Now it's 13, and Alnylam was one of them. So and probably these multi-analyte signatures are something that they may be pursuing internally, but not publishing on. And in fact, I will say it's pretty rare that a team within a pharmaceutical company is able to publish on findings like this. I was very excited to see that they took the time to put it out into the literature to help others understand this mechanism.
Well, like we were talking about how difficult it is to assess the diagnosis, then one thought, right, is that the important findings is neurofilament light as a diagnostic marker. Is that right?
Yeah.
Yeah, exactly. As a diagnostic marker and one that can, monitor disease progression and response to therapy, right?
So what do you mean by? I'll also say that go ahead.
Paul Nioi, who was the principal investigator on this paper, the final author, has a long history with Decode and Amgen, very, very strong genetics specialist. He's quite accomplished as a scientist. And then Samina Ticau is a principal scientist, of course, at Alnylam.
And as far as then you mentioned disease progression, what do you mean by that? So if I understood correctly, the people with disease with placebo, right, they had this rising level of NFL, and people that were on the medication, right, stayed constant. What will a biomarker do with regard to telling how far you are coming along with the disease? You mentioned disease progression.
Actually, as far as I understand, it's not like a constant. I think that it's also decreasing and reaching out to the levels of the healthy controls. And that is because they are running the healthy controls, right? And you see that initially healthy controls with the disease, they are like 4 times lower, roughly. Mm-hmm. And by having the patisiran, right, they see that levels of the patients, in the patient samples, the levels of NFL goes leans towards the healthy conditions, right? That was the case. And for this, it was responsive to the therapy at the end, right? I think that was a really nice thing with this protein.
I see.
Yeah, and I think
if I understood correct this topic, actually.
Yeah, I agree. I think that the levels trended toward healthy samples in the patisiran treated, and they reached a level that they stayed at from 9 months to 18 months. At least that's what it looks like in the significance levels and the graphs. That essentially they're saying, I think, that they're able to have a quick response, and that those patients appear to stay steady at least over those 18 months, right? But that longer studies are needed to really nail down the value of that as a marker over that long period of time.
Now, if I understand correctly, Sarantis, you mentioned this patisiran drug is actually double-stranded RNA. It's silencing these particular transthyretin genes that are aberrantly mutated, what have you. Is it just one dose? Do they have to take multiple doses?
This I don't know, and actually
maybe
I think it's one dose. They are injectable, and it happens to the liver, it's targeted to the liver. But I don't know if there's a second dose or third dose on this. But I think it's intravenous and targets liver.
Yeah,
liver
I see.
tissue.
Thinking about it, it probably does make sense that they're a regular dosage. I was thinking about sort of after the trial was over, right? They used the broadest panel that they could with the Olink Target 96, the 13 or 14 of them. But that was for discovering more, right? Wasn't the goal of that to look at mechanism of action in terms of how patisiran was working?
I think it's not like how patisiran is working. It's like how the silencing of the gene affects other proteins that can be potential biomarkers.
I see.
Because at the end, patisiran targets the gene TTR gene. Doesn't target any other gene, targets the gene. Then what we are screening, what they actually are screening, is the effect on other proteins that are made connected upon, this amyloidosis.
Got it. Well, the systems biology of it gets pretty complicated because you're just talking about reducing the expression of one gene, but we're a system. So,
But we can speculate what they're doing internally, but we're not going to know if they're not publishing on it, right?
Yeah.
Absolutely.
Sure.
Absolutely.
Well, Cindy, you mentioned some interesting things related to multiple sclerosis. And, with neurological disorders, I mean, coming from the genomics perspective, I myself know very little about neurological disease like multiple sclerosis. But you mentioned a conversation about Epstein Barr Virus and multiple sclerosis. Do you mind sharing that?
Yeah, sure. So I think it's quite the buzz in the Twitterverse, and several people I trust have been really impressed with this paper, and we can put it in the show notes, around really assigning causality to a subset of MS patients. And MS is, of course, very different from hereditary transthyretin-amyloidosis. It's a autoimmune disease, but that Epstein Barr has been identified causally as causing multiple sclerosis in a subset of those patients, which, that's been a postulate in Parkinson's disease, of course, as well, that virus can turn on something that's sort of hidden in your genome and make it active. In that case, I haven't done much reading on Parkinson's disease or where we're at with that hypothesis, but I certainly know people with Parkinson's disease who can tell me stories about having had a viral infection very soon before very recently in the past, before their onset of symptoms. So it's a compelling idea, and understanding the mechanism of that would be incredibly important.
Now, Epstein Barr virus, isn't this one of those viruses that's endemic in the population? Meaning it's very, very common.
Like 90% or something. Let's look that up.
Okay. And if you have Epstein Barr virus, it might present itself as a mild cold or something, right? You wouldn't know that you had Epstein Barr virus. But what you're saying is people that get, or maybe I'll put it this way, there is a subset of multiple sclerosis patients who are triggered by getting infected with EBV. Is that correct?
That's right. Yep, where they could find the link to EBV infection. Wow. So the numbers I'm seeing from Cleveland Clinic are an estimated 50% of all children that are up to 5 years in age have been exposed to EBV, and 95% of adults experience an EBV infection in their lifetime.
Okay, and they experience an infection, they recover like from any other virus, right? And they go on with their life, but then there's that subset of, so they don't know what the mechanism.
Long COVID, right? It's
they don't know what the mechanism is, right? Because we're talking about a complex autoimmune disorder, and we're getting into the nuances of autoimmunity, right?
Yeah, exactly. And I think, I've heard it linked, although I don't know the scientific case for it, but I've heard people claim that it's linked to fibromyalgia, or what's the other disease that's been compared to long COVID? Mike Snyder is doing a study, I think, looking, or he proposed a study looking at the difference between chronic fatigue, CFS, and long COVID, just to understand what are the differences. And it might be interesting to see what the link.
Well, getting back to the EBV and multiple sclerosis story, is there any development preventative that could prevent MS for these subset of people?
Yeah, I'm not. I said, I haven't read that paper directly. I just know it's quite the buzz from people that I trust.
Yeah, interesting, man. And then you mentioned that EBV may be implicated in some of these other neurological disorders, but if 95% of them of adults being exposed, what can you do, right?
Yeah.
Do we go
exactly.
back to wearing masks all the time? No, I'm not suggesting that. I'm just thinking about the sort of practical implications. So.
Well, Epstein Barr, remember, is a herpes virus, right? So those are very hard to avoid.
Yeah. This battle, right, between infectious organisms and humans.
That's right.
It's been going on for a while.
That's right.
Well, Sarantis, you want to close with any thoughts?
No, I think the thoughts and the nice take-home message for this type of papers is like, we hope we're going to see double-stranded RNA therapies coming. We are in the RNA era, and therapies from vaccines to double-stranded RNA, and the proteomics can really be a nice tool to assess biomarkers, right? Like endpoint effects. So that's pretty much what I say and what excited me in this paper.
That's great. Well, thank you for the time today, friends and co-hosts.
Thank you very much.
Okay.
Yeah.
Thank you very
for
much.
joining.
Bye.
Bye.
Bye. Bye.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.